Welcome, listeners. So, in this video, I'm going to talk about primary and secondary structure of proteins. The structure of proteins is of much importance, not only just for GATE biotechnology examination, but also for GATE life sciences, for CUET PG entrance, for CSIR JRF, for ICMR JRF, for DBD JRF, and many other kinds of higher level of exams. And I try to make every concept clear to all of you. So I think you will enjoy the video, and I appreciate if you come up and write a few lines in the comment section that how you feel my videos. Yeah, how you feel about my videos. Thank you so much.
Viewers. So, we will now turn to polymers of amino acids, and that are peptides and the proteins. Peptides, which are formed by the combination of minimum 2 amino acid residues. When the hydrogen atom of amine group of one amino acid eliminates with the hydroxyl group of the COOH group of the other amino acid, it eliminates in the form of water, leaving a bridge between them, a CONH group which is also a amide group, forming a bond termed as peptide bond. So, a peptide bond is formed by the condensation reaction occurs between minimum 2 amino acid residues. When 2 amino acids join together, it results in the formation of a dipeptide having one peptide bond. And when 3 amino acids join together, it results in the formation of tripeptide having 2 peptide bonds. Don't ever confuse with the dry and tie term with respect to peptide bond, because dipeptide contains one peptide bond having 2 amino acid residues. So as in the case of amino acids, it also has an amino terminal and a carboxyl terminal. Now, the peptides range in length from 2 to 10,000, and in that case they are termed as dipeptide, tripeptide, oligo and polypeptide. Polypeptides, formerly called as proteins, which consist of a single peptide chain, while others have many chains called multi-subunit proteins. The individual polypeptide chain in a protein can be same or different. If at least 2 chains are same, they are said to be oligomeric, and the identical units are referred as protomers. So naturally occurring peptides are 2 to thousands of amino acid residues long. Let's have some examples of the short peptides as well as the larger ones. A few examples of short ones are oxytocin and bradykinin, which contains only 9 amino acid residues, while thyrotropin releasing factor contains 3 amino acid residues. Example of slightly larger polypeptides are insulin, which contains 2 polypeptide chain having 30 and 21 amino acids in each chain. Glucagon, which contains 29 amino acids, and so on. Do you know you can synthesize a small peptide commercially, and the best example of it is artificial sweetener or aspartame, or formerly called as NutraSweet, which is a dipeptide of aspartic acid and phenylalanine. So by knowing the molecular weight of a protein, you can calculate the approximate number of amino acid residues it contains. The average molecular weight of 20 amino acid is around 138, but since most of the proteins contain smaller amino acids, so in that case the average molecular weight of 20 amino acids would be 128. And since formation of a dipeptide results by elimination of a molecule of water having a molecular weight of 18, that's why reduction of 18 from 128 gives rise to 110, and division of the molecular weight of a protein by 110 give rise to an idea of approximate number of amino acid residues a protein contains. As like the case of carbohydrates, the protein can also be associated with non-protein moieties. These can be lipid groups, these can be carbohydrate groups, there can be phosphate, there can be heme, there can be flavoproteins, and there can be association of some metals inside the proteins. All these extra moieties or non-protein elements are known as prosthetic group. The common example of this is heme protein, which contains iron in the form of porphyrin ring, and the common example of it is hemoglobin and myoglobin, and we will discuss both the proteins in our next lecture. So, the credit of the overall three-dimensional structure of protein or the careful analysis of peptide bond goes to Linus Pauling and Robert Corey, who laid the foundation of present study of protein structure by X-ray diffraction analysis. He said, or he elucidated, that all the 6 atoms in a peptide group lie in a single plane, means you can draw all these atoms on a single paper, with oxygen atom and hydrogen atom are in transposition to each other, while the alkyl groups of both the amino acids are also in transposition to avoid a steric hindrance. Let's have a look at the alpha carbon of both the amino acids, which are apart by 3 covalent bonds, C alpha C, NC alpha and NC alpha. Okay. So, the alpha carbon of the adjacent amino acid residues are separated by 3 covalent bonds. But we have to discuss the peptide bond right here, the ping bond. So, he found that the peptide CN bond is shorter than the CN bond found in any other moiety. Why? This is because of the partial sharing of electron between carbon and nitrogen, which gives it a partial double bond character or a resonance-like structure. So, the length of the double bond becomes shorter as compared to the single peptide bond, and always remember that the rotation of the atoms around double bond is always restricted, means peptide CN bonds are unable to rotate freely because of the partial double bond character. This is what it gives a planar and rigid structure having a high activation energy. High activation energy means not easy to break, and in biological system their half-lives are around 7 years. So, the peptide bond which we have discussed here are in a straight line, right? But in biological system they are present at some angles, and these angles are referred as phi and psi, where psi is the angle between the bond between C alpha and C carbon, this region, and phi is the bond angle which forms between the N and C alpha carbon. So, when you plot phi against psi, you got a plot which is termed as Ramachandran plot, given by a great scientist G.N. Ramachandran. Why this plot is formed? To give you an idea about the conformations of the protein which are allowed by the biological system and which are strictly not allowed. For a reference, when the polypeptide chain is in fully extended conformation, these angles are 180 degrees. Okay, so in principle phi and psi have any values between -180 to +180 degree. Let's take an example of L-alanine, on which the plot is based on. You will see some dark blue areas right here, some medium blue areas right here, and some light blue areas. The dark blue areas represent all the conformations of proteins which are easily possible in the biological system, while the medium blue areas represent the conformation which are formed at the extreme limits of unfavorable atomic contacts, while the lightest blue area are the conformations of protein that are permissible with a little flexibility in the bond angles phi and psi. So we will get a better idea of this plot when we will discuss few structures of the protein. So as we discussed that peptide CN bond is unable to rotate freely, but the protein contains a number of covalent bonds. So there can be a number of rotations, so there can be unlimited number of conformations. I just want to say that it is not only peptide bond which forms proteins, but there are several other covalent bonds which give rise to a protein structure. And because of these bonds, there can be unlimited number of conformations. But in biological system, only one or generally few dominates, which are having lowest Gibbs free energy. So proteins in any of their functional or folded conformations are called native proteins, and the ongoing slides are based on the four themes. First, the three-dimensional structure of a protein is determined by its amino acid sequence, and as I told repeatedly that in biological system the structure determines its function, so it is the second point. The third one is an isolated protein has a unique structure, and the fourth and the most important point is the forces stabilizing the protein structures are weak interactions, not the covalent ones. So biochemists have distinguished several levels of protein structures, and they are primary, secondary, tertiary and quaternary, and we will discuss each of them in very details. Starting from the primary structure, which is a simple and linear arrangement of amino acid residues by a covalent bond. If you take an example of bead thread, the beads here represent the amino acids and the thread represent the covalent bonds. So primary structure is the simple linear structure of amino acid residues joined mainly by dipeptide bonds and sometimes by disulfide bonds. The most important element of primary structure is the sequence of its amino acid residues. Secondary structure results when there is some folding in the primary structure. It refers to a particularly stable arrangement of amino acid residues giving rise to recurring structural patterns. Recurring means same. So secondary structure refers to folding of a polypeptide backbone which sometimes gives origin to a few stable conformations, and alpha helix and beta sheets are among them. Let's discuss alpha helix. When you fold a primary structure around an imaginary axis, a helix formation results which we termed as alpha helix. In biological system, only right-handed helix is favored. When you see in details, you will find that the R groups, the bulky R groups protrude outward from the helix. It contains 3.6 residues per turn and the length of the groove is 5.4 angstrom. The typical length of alpha helix is 10 amino acid, but it can range from few plus to few minus. So coming to the structural stability of the alpha helix or the first secondary structure, which are stabilized by hydrogen bonds, and these hydrogen bonds are formed between first and fourth amino acid residues, as it contains 3.6 residues per turn. So first amino acid hydrogen bonds with the fourth amino acid, and in this way the stability is provided to the alpha helix. Like in the case of dipeptide and tripeptides, it also has one positive or amino terminus and one negative or carboxy terminus. So taking chemistry into mind, near positive end negative charged amino acid groups can be found, and near the negative end positive charged amino acids are found. Okay, so the most favored amino acid composition of alpha helix are methionine, alanine, leucine, glutamate and lysine. Proline and glycine are strictly not found in the alpha helix because of the unique structure of the protein. It provides a destabilizing nick if found in between the alpha helix. However, it can be found as a first amino acid. Glycine which has a more conformation flexibility, if it is found, it results in the formation of coiled structure, which is different from alpha helix. Same charged amino acids, if placed adjacent to each other, repel each other. So if they are found, they are 3 residues away, and aromatic amino acid residues are often similarly spaced, resulting in hydrophobic interaction. Bulky amino acids like asparagine, serine, threonine and leucine also destabilizes the structure of alpha helix. So you can have an idea that how important is the amino acid composition in determining secondary structure of the protein. Let's discuss the another one, the beta sheet, in which the backbone of a polypeptide chain folds into a zigzag manner, giving it a more extended conformation. These are also stabilized by hydrogen bonds, and these bonds are formed between the 2 strands. If the run of 2 strands is on the same side, means same carboxy to amino terminal, beta sheets can be parallel in nature, and if 2 beta sheets run in the opposite direction in terms of amino to carboxyl end, they are anti-parallel. And there is a difference between the repeat period in the parallel and anti-parallel strands. For having repeat period is shorter for parallel conformations as compared to the anti-parallel. The red moieties right here are the R groups, and the point to be noted here is that in the interface between the 2 strands, the R groups cannot be bulky. So smaller R groups are favored to stabilize the structure of beta sheets. Let's have in details the amino acid composition which are favored to form beta sheet, and these are tyrosine, phenylalanine, tryptophan, and some beta branched amino acids, threonine, valine and isoleucine. Yes, proline can be found in this structure when present on the edges. So this is the difference between alpha helix and beta sheets, and it's a rod-like structure, while beta extended into a zigzag manner, and it can be clockwise or anti-clockwise, and beta can be parallel or anti-parallel. So coming rise to beta turn, which results in globular proteins where polypeptide chain reverses its direction. These are the polypeptide chain, and here the direction is reverse. This is the polypeptide chain, and here the direction is reverse. So when reversal in the direction results, there is formation of beta turn, and it's a 180-degree turn involving 4 amino acid residues, in which hydrogen bond forms between first and the 4 amino acid residues, while the second and third does not participate in hydrogen bonding. It can be type 1 and type 2, where glycine is the 3rd amino acid always in the case of type 2, but in biological system type 1 is mostly found. We will discuss the amino acid composition of beta turn in detail by taking an example of the most commonly found amino acid, proline, which is actually found in the case of beta turn. And the point to be noted here is that in biological system, all the amino acids are present in trans configuration, where the functional groups are in the opposite in direction to each other. But in beta turn, proline specifically is found in cis direction, where the functional group present on the same direction. Why? To allow or to give the structure a turn-like form. You can see a beautiful turn is formed when trans is converted into cis. So this is the cis and trans isomers of proline, and the proline, which was not found or very less found in the alpha helix and beta sheet, is mostly found in the turns. Now a less common gamma turn is also found in biological system, which is a 3 residue turn with the hydrogen bond between first and third residues. Let's plot all these structures onto the Ramachandran plot to give you a better idea why we have plotted or why we have discussed the Ramachandran plot. Here the anti-parallel beta sheets, the parallel ones, the right-handed beta sheets and the right-handed alpha helix you can see on the plot, means these all are favorable conformations. You will also find left-handed alpha helix here, which is theoretically possible, but in biological system it is not possible. So this is all about primary and secondary structure of a protein. We will discuss tertiary and quaternary structure in the next video. But before leaving, let's discuss a few questions. The peptide bond is not partially double bonded, polar, planar or trans in proteins, and the correct answer is it cannot be polar in nature. The second one, the forces that maintain the three-dimensional structure of a protein is mainly, yes, they are non-covalent interactions, and we have already discussed it. The mean molecular weight of an amino acid in a typical globular protein is, and we have calculated it as a 110. Which of the following amino acid stabilizes alpha helix structure in the proteins? Always remember, it is the hydrogen bonds which stabilizes the secondary structure of the proteins. And